Welcome to the second installment of the QBI Weizmann Institute Symposium on Molecular Systems Biology, co-organized by myself and Tanja and Saki, Noam and Danny. We have an equally exciting day today as we did last week. These were the speakers last Wednesday, and we have a set of speakers today, both from QBI UCSF and the Weizmann Institute. And today, Noam and Tanja are going to be chairing the two sessions, which are going to be split up by a break. And the last thing I just want to say is for next week, the PIs are going to get together for a discussion on how to go forward scientifically and some of the efforts around fundraising. And listed here are just the PIs that are speaking, but we're, of course, welcome to any other PIs at both institutions that would like to participate in next week's discussion. And just to remind the speakers, at 8 minutes, you're going to see a specific mark. Carolina, can you show that mark? There's 10-minute talks plus 5-minute questions. So at the 8-minute mark, you will see the following. Very subtle red dot, denoting you have 2 minutes left. So with that, I'll turn it over to Noam and Tanja to start the session.
Hi everyone. So it's really exciting to initiate the second meeting we have of UCSF in Weizmann. For me, it's like a full circle because until 6 years ago, I was actually a postdoc in UCSF. So it's really exciting, and I'm sure we'll have an exciting talks today. And I'll present the first speaker, which is, I'll move to the next chair, Tanja, to say a few words, and then we go for the first session.
I don't have much to add. Welcome, of course, everybody. I really enjoyed last week's session. I learned a lot. It was really exciting science. And I'm sure we'll have the same this time. So I'll hand it back to Noam.
Right, so I'll introduce the first speaker for today, Natalia Jua from QBI UCSF, which is Associate Professor, and the title is Molecular Mechanism of Receptor Kinase Signaling. Welcome, Natalia.
Thank you so much. Thank you to all the organizers for giving me an opportunity to speak. Can everyone hear me?
Yep.
All good? Okay. So my lab focuses on signal transduction, and in particular, the molecular machines that are responsible for the mitogenic signals, which by and large are the single-pass transmembrane receptor kinases. There are two types of receptor kinases, general classes, receptor tyrosine kinases, which we are looking at in the lab since the beginning. And our most recent interest is on a second class, receptor serine threonine kinases, which consists of TGF-beta BMP receptors. And as Yaron has beautifully outlined last week, the combinatorial power of this family of receptor ligands is quite remarkable. We're interested in it on the receptor side and how that combinatorial power is established at the level of mechanism from the perspective of the receptor. And all these receptors can be divided into two types, type 1 and type 2. And the way the signaling works is that two type 1 receptors come together with two type 2 receptors, forming these tetramers, which are induced by binding of dimeric ligands. And this tetrameric arrangement is really unique to this class of receptors because in a receptor tyrosine kinases, it's really the dimer that is this basic activation unit. We know that it's a tetrameric arrangement because there are multiple structures of the ectodomain modules, and you can see here a type 1 and type 2 ectodomains sort of scaffolded around this large dimeric ligands, but there are no structures of any complexes between type 1 and type 2 kinases, so the intracellular modules. So we have no idea how mechanistically activation occurs. we know something, and we know that the very important component of activation is a phosphorylation of the GS domain that's present on type 1 kinases. This domain is phosphorylated by type 2 receptors, and that's essential for signaling. And the reason why this is essential is because phosphorylation removes the inhibition that the GS domain sets on type 1 kinase by binding to it. This is additionally stabilized by this FKB inhibitory molecules. So phosphorylation is essential. But how it really occurs, why do we need a tetramer? We really don't know. So we thought we're going to take a look because this was mechanistically very interesting. And we focused on this complex between ALK2 type 1 receptor and BMPR2 receptor. This work was done by Jennifer and Chris in my lab in collaboration with Akiko Hata here at UCSF. We chose these receptors because they carry very interesting disease mutations. In particular, BMPR2 has inactivating mutations which are localized on the surface of the kinase domain, so far away from the active site, and they were really mechanistically unexplained. We purified kinase domains corresponding to these receptors, and when we mix them in solution, right away we noticed that they form a complex, which you can see here as this black lane. And through a variety of assays, we realized that it's a heterodimer. So it's not a tetramer, it was a heterodimer. We were intrigued and we wanted to characterize it. Crystallography didn't really work out, so we used an integrative approach in collaboration with Yiping and Palin at DE Show Research, where we used long-time molecular dynamics simulations to look at stable complexes that these kinases would like to form. And we sort of narrowed down on two complexes that we really, really liked. In one case, the kinases were interacting through the N-terminal domains, which we called N-lobe dimer, and in our case, through C-terminal domains. We really preferred the N-lobe dimer right from the get-go because the way type 2 BMPR2 kinase was sitting on ALK2 really was incompatible with the autoinhibited state, and we thought this dimer would really nicely explain how type 2 activates type 1. So we decided to test it by using a hydrogen deuterium exchange mass spectrometry in collaboration with Eric Underbaki from Iowa. And when we did this experiment, we noticed that the protection that we see on these kinases in solution is not in the N-lobes, which would be indicative of their interaction via N-lobes, but it's in the C-terminal lobes, which really was consistent with the C-lobe dimer. So then we thought, you know, at least in solution, these kinases must be interacting through the C-lobe interface. Kind of made sense when we looked at this interface. There was this complementary electrostatic surface potential on ALK2 and BMPR2. And when we made mutations in this interface based on our MD model, basically all of these mutations disrupted formation of the complex in solution, which reassured us that is what probably is happening in solution. But what this dimer means, does it have any function? None of these mutations affected catalytic activity of these kinases, but we noticed that a majority of these mutations disrupted signaling by ALK2 and BMPR2 in cells induced by binding of BMP4 ligand. Most of these mutations those that didn't were the mutations that were sort of at the periphery of the interface. So we thought it's interesting. This dimer seems to be important for signaling in a way that's not directly related to kinase activity. And another piece of evidence that really convinced us about this dimer was the fact that on the BMPR2 part of the interface, a lot of mutations that are observed in pulmonary arterial hypertension has clustered. And those are the surface mutations I told you about earlier. And our model really nicely explained how they could disrupt the interactions at the interface. And introducing them point by point, single point mutations completely dissolved this dimer in solution and blocked signaling in cells. So the interface is important, but what is the role of the C-lobe dimer? So, you know, all our data is consistent with the importance of this interface in activation. But when we looked at it, we thought this dimer really looks like an autoinhibited complex, because what's happening here is the active site of type 2 kinase, BMPR2, is just too far from the GS domain. So this complex cannot achieve the most important thing, which is phosphorylation of the GS domain. On the contrary, it actually prevents it by sequestering the GS domain away from the active site. And then we reminded ourselves that, you know, we have to think about a tetramer. So what if we put two of those dimers together? Can we even do that? And turns out that the best way to do it is to connect them via this N-terminal dimer interface that we have also identified in MD simulations. And when we do that, what you see here on the right in the cartoon, now the GS domain of ALK2 is perfectly and snugly positioned to access the active site of the BMPR2 kinase. This is a molecular dynamics model of this heterotetrameric complex. You see the GS domain beautifully fits in the active site of BMPR2, and that's happening now across this N-lobe dimer interface. And when we make mutations, which are also shown here as dots, but purple, in this interface, we again block signaling. So how does this all come together? What did we learn? We think that this dimer we see in solution is quite stable, and it might actually even form between the receptors in the absence of ligand binding. And heterodimeric complexes between type 1 and type 2 receptors have been observed in the absence of a ligand, so that would be consistent. So this dimer is inhibitory, but at the same time, it primes the complex for activation, because now when the ligand binds, this lower affinity N-lobe dimer can form and perfectly position GS domain for phosphorylation. So going back to the systems level and connecting it to Yaron's talk from last week, is this something specific to ALK2 or BMPR2, or is it a universal way by which these receptors form tetramers? And at this point, we really don't know. We can form complexes between BMPR2 kinase and also ALK3 and ALK6, which are other type 1 kinases, which is encouraging. And just this last slide shows you how conserved, at least, is the electrostatic surface potential on type 1 kinases and type 2 kinases in this region, suggesting that it's possible that these tetramers form across the family. But of course, how is this regulated? How different ligands regulated is a big and very interesting question. So I'll end right there and just acknowledge my lab, our collaborators and our funding. Thank you, and I'm happy to take any questions.
Thank you, Natalia. Are there any questions from the audience? So I have a question. So you talked in the beginning about mutations that are seen in patients. So how are these affecting the surfaces you discovered?
Yeah, so these pulmonary arterial hypertension mutations are right at the interface on the BMPR2 side. I want to note that there are many different types of PAH mutations. Not all of them cluster in this interface. This receptor has very many clever ways by which it can inactivate its signaling. But these unexplained type of mutations, which were surface mutations in the kinase domain, at least are very nicely explained by this model. The other mutations on the ALK2 side are actually activating mutations, and what they do is basically they dislodge the GS domain and sort of promote the interaction between GS domain of type 1 kinase and the type 2 kinase.
So there is another question. First, very neat talk. How does the ligand binding switch from dimer to tetramer?
You know, those are very big questions, and I don't think anybody understands. I think that, for many different ligands, and there are subtle differences between TGF-beta receptors and BMP receptors, a ligand can initiate a dimer formation and then proceeds into a tetramer. And by dimer, I mean type 1 or type 2 receptors are first clusters on the dimer scaffold, and then the other type is recruited. What I was referring to is formation of heterodimers in the absence of a ligand, which has been observed as well. as some sort of, primed inhibited complexes, which are sort of at the membrane, waiting to be activated by ligands. And, evidence for it is from imaging, single molecule, sort of tracking, diffusion studies on cells. Nothing really biophysical that would address this question very well. And I think this, that's a huge opportunity to really understand that system in the future.
Any more questions?
Well, maybe I can ask one more. So your model, Natalia, would predict that mutations in the N-lobe interface also have functional effects, right?
Yeah, and I showed these mutations we've done at the tetramer interface. They were done also on the N-lobe dimer interface. You have to be careful with those mutations because it's very easy to activate the kinase by mutating the N-lobe because you just dislodge the GS domain. So we had to very cleverly design them that they will keep that interaction, but really just disrupt the tetramer to understand whether this tetramer is important for activation.
So there is another question. Have you noticed any allosteric effect of liminalization from the HDX MS?
Any other interfaces? Is that the question? This is a very good question. And we saw some protection at the N-lobe interface, which was very interesting. And we saw it higher when we had the GS domain present, which we couldn't really understand. Those signals, though, are not very, very strong like they were at the C-lobe interface. So I don't know if it's indicative of some transient tetramer formations in solution. My understanding is that this tetramer really only forms stably in the context of full-length receptor when you have stabilization of the complex with a ligand. But perhaps that's what HDX was telling us as well.
Right, thank you, Natalia.
Thank you.
And we move on to the next speaker, Professor Noam Stern-Ginossar from the Department of Molecular Genetics, which will tell us about the immune system of bacteria. Apparently, there are things beyond CRISPR.
Apparently so.
Gita Navlika from QBI, from the biochemistry and biophysics at UCSF, and she will talk about can phase separation explain heterochromatin properties. Looking forward.
Thank you, Noam, and thank you to the organizers for inviting me to this symposium. So our lab is interested in understanding how chromatin structure and organization regulate genome function. And we largely use biochemical and biophysical approaches to address these questions. Today, I'm going to share some of our recent work on understanding how heterochromatin is formed, and to give you a flavor for how we think and the ways in which we ask questions. Move the slides. There we go. Okay, so here's a classic way to visualize heterochromatin. What I'm showing you is a mammalian cell where the DNA in the nucleus is stained. The darkly stained regions shown here are heterochromatin and comprise of condensed regions of the genome. And the lightly stained regions here are euchromatin and comprise of more open regions of the genome. Euchromatin has more active genes, whereas heterochromatin has genes that are more often silenced. Now, in addition to silencing, heterochromatin also causes repression of recombination. It is terribly important for chromosome segregation, and it somehow confers rigidity to the whole nucleus. Now, one of the most highly conserved types of heterochromatin is mediated by this protein HB1 that has this unusual architecture. It's more often than not a dimer. It has domains that directly bind histone tails, shown here by the chromodomain. It has unstructured regions that directly bind DNA and RNA, shown here by the positively charged Hind region. And they also have dimerization domains, shown here by the chromoshadow domain. Now, this type of heterochromatin mediated by HB1 is conserved all the way from yeast to humans. This type of heterochromatin also has been reported to have some apparently paradoxical properties in cells. So on the one hand, heterochromatin shown by these kind of stable domains can be maintained on the order of hours without disassembly and in the same place in the nuclear architecture. So these compartments of heterochromatin can be stable on the order of hours on one hand. But on the other hand, if you take fluorescently labeled HP1 molecules and carry out FRAP analysis, these molecules shown here in these older studies, if you FRAP them, they recover on the order of seconds. So somehow compartments that are stable on the order of days, shown by these static pictures, are maintained by core heterochromatin molecules like HP1 that are dynamic on the order of seconds. Okay. So these are the kinds of questions we're trying to understand from the perspective of the underlying biophysics. So what are the biophysical mechanisms underlying these types of properties of HP1 mediated heterochromatin? Now, we've been studying HP1 mediated heterochromatin for several years, and we've learned a few things that I'm summarizing in this one slide. What we've learned is that HP1 molecules can oligomerize across chromatin, and shown here are the basic units of chromatin and nucleosome, along with the histone tails. These HP1 molecules can oligomerize, and when they reach a certain density on chromatin, we found that these molecules can also phase separate. And phase separation by HP1 molecules results to formation of these kinds of droplets that can enclose the underlying chromatin within them. Now, this kind of phase separation provides a very attractive mechanism to explain how heterochromatin can enclose and compartmentalize large regions of the genome. And since these discoveries that we made a few years ago, we've been trying to better understand what does phase separation, what confer on heterochromatin, and are there properties that phase separation confers that can help us explain how heterochromatin acts in cells. And so today I'm going to share with you the work of a brilliant and talented graduate student, Madeleine Keenan, who's a joint graduate student between my lab and the lab of Cy Redding. So everything I'm going to tell you for the rest of the talk is a complete collaboration with Cy's lab and my lab, and has been spearheaded by Madeleine Keenan, a joint graduate student. What Madeleine was interested in is asking is, what types of mesoscale properties can be conferred by phase separation of HP1 molecules? And to address that, she did a bunch of experiments. I'm going to only share a subset of the experiments that she did. And the kinds of experiments she did was, she said, okay, let me go to a simplified system. Let me use phase separated droplets formed between HP1 molecules. And shown here, she's going to use the main HP1 molecule in humans, HP1 alpha. And interaction of HP1 alpha with a large piece of DNA can result in phase separated droplets. And so she decided to use these droplets as simple models for HP1 DNA territories. And underlying all her questions were trying to get a better understanding of how dynamic HP1 binding results in stable and regulatable genome organization. So one of the first questions she asked using the simple model system was, when you make these kinds of territories in a test tube, how quickly does HP1 alpha exchange between the droplets? And to do this experiment, she made two types of droplets, green droplets and red droplets. The green droplets had a green labeled, fluorescently labeled HP1 alpha with unlabeled DNA. The red droplets had a red fluorescently labeled HP1 alpha with unlabeled DNA. What she did was she mixed these two preformed droplets and asked, when the droplets fuse, does the HP1 alpha molecule, do the HP1 alpha molecules in each of these droplets mix up with each other? So do the HP1 molecules mix when the two droplets fuse? Now, when the droplets fuse, if the HP1 molecules mix, you expect all the droplets to be yellow. Okay. So that's what she saw. She mixed the droplets, waited for an hour and a half, and all the droplets shown here in the combined channel are yellow, indicating that in the time scale of this experiment, when the two droplets fuse, and shown here are two droplets in the act of fusing, even as they're in the act of fusing, the two types of HP1 molecules, the green and the red, have completely mixed up. And so you only get yellow droplets. She then did the converse experiment, where she now, again, the same identical components, but now she labeled the DNA. Okay, so we have green droplets again and red droplets again, but now the DNA is labeled. And she asked, when the droplets fuse, can the DNA exchange between the droplets? Again, the same process. She made green droplets, red droplets, mixed them, and waited for an hour and a half. And this time, she got a very different kind of result. You could see the red in the droplets, you could see the green in the droplets, but even in the droplets that have fused, shown here by one example of a fused droplet, where you have fusion between a red droplet and a green droplet, the DNA remains in its territories. So even though the two types of droplets have fused, the DNA has a hard time mixing up. So how can we explain this somewhat unexpected result? Well, again, we know a little bit about how this molecule functions, and we know that this molecule can oligomerize, and it can bridge across multiple parts of DNA. And so what we propose is that multiple weakly bound HPO and alpha oligomers can bridge different regions of DNA and kinetically trap the DNA in its own territory. And this would also result in increasing the effective viscosity of the large DNA molecules. But at the same time, individual HPO molecules, because they come on and off on the order of seconds, can rapidly come on and off and explore the surrounding territory, can mix between the neighboring droplets or the neighboring territories. But at any time, the piece of DNA is always occupied and always held down with multiple HPO molecules. Okay, so if this is how we are imagining HPO and DNA territories, and if these are the kinds of interactions that are holding these territories in place, can we get an understanding of how stable these territories are? So to get a sense of how the mechanical stability of these territories, Madeline teamed up with a postdoctoral fellow, Lucy Brennan, who's a joint postdoc between Cy Redding's lab and Bassem Al-Sadi's lab. And what they did together was to do an optical trap experiment, where they took a long piece of DNA, lambda DNA, and they attached both ends of this lambda DNA to two ends of an optical trap, either side with a polystyrene bead. Each bead is held stationary by an optical trap. And they asked, what happens when we assemble a droplet in the middle of this piece of DNA and pull? Okay. Now, if these molecules are coming on and off on the order of seconds, what they expected was if you pull on one end on one bead, the droplet would fall apart because these droplets are liquid-like. Okay. But instead, this is what they saw. And I'm not going to show you all the pulls, but they did 10 successive pulls. So shown here is the droplet made from fluorescently labeled HPO and alpha. That's how you can see the droplet. One bead is being pulled. It's pulled 10 times successively, and yet the droplet does not fall apart. You can do force extension curves. And what they found when you do force extension curves is that while naked DNA in the absence of droplets behaves as you would expect DNA to behave when you increase the force, when you have a droplet formed in the middle of the DNA can't extend beyond a particular region, a particular length, and the droplet remains intact. And more importantly, the droplet can withstand up to 40 piconewtons of force. So how do we put this in context? RNA polymerase stalls at 20 piconewtons. Okay. So somehow these structures that are formed with HPO and alpha in DNA are stable up to 40 piconewtons of force. And you can imagine how they might be resistant to the brute force action of RNA polymerase, and perhaps one way by which these structures resist transcription. Now, if these structures are so stable, how might they be regulated in a cell? Here, Madeline turned to ask whether other paralogs of HP1 and so can regulate the structures formed between HPO and alpha and DNA. Now, everything I've told you so far is in the context of a major form of HPO and alpha in humans. There are other paralogs such as HPO and beta that do not form droplets of DNA and do not compact DNA. And so what Madeline did was she asked, can HPO and beta act as a poison for the droplets formed between HPO and alpha and DNA? And shown here are droplets formed with HPO and alpha with increasing concentrations of beta. And what she found is that beta can dissolve the HPO droplets pretty readily and very quickly. And so we think HPO and beta acts as a poison by heterodimerizing with HPO and alpha. So to bring all of this together in the conclusion slide, what we think so far is that oligomerization of HPO and alpha and the rapid on-off dynamics of this molecule, together with their ability to form phase-separated droplets, can confer pretty biologically useful properties. Okay? So shown here are just 3 of these properties. These kinds of biophysical properties can give you DNA compartments that inhibit mixing of DNA but allow mixing of HP1. And this could help explain how you might enable repression of transcription and recombination. The compacted DNA is highly resistant to biologically relevant forces. And this kind of mechanical rigidity can explain how heterochromatin structures can confer rigidity to the whole nucleus. And finally, because these structures are maintained by molecules that come on and off on the order of seconds, there's also a potential to rapidly dissolve these structures by invading molecules that compete with HP1 molecules. Now, I want to end by saying that while all of these data are in the context of HP1 molecules, there's nothing special about the types of properties that I've described here. And we imagine that there are other molecules that organize the genome that might share some of the properties that we are finding for HP1 alpha molecules. And these may reflect some general principles by which phase separation mechanisms might contribute to genome organization. So I want to end just by thanking again the folks who did the work. Madeline Keenan really is a champion of this work. I'm really thankful to Cy Redding for being such a wonderful co-conspirator on this project. as well as Adam Larson, who got us into phase separation in the first place, and Srina Sanoli, another joint postdoc who's done amazing work in phase separation with chromatin. Thank you.
Thank you, really fascinating. I see we already have a question. I'm lost in the questions.
Well, maybe I can ask her. This is really beautiful. Are there other poisons, such as perhaps post-translational modifications?
Absolutely, absolutely. Natalia mentioned phosphorylation. So HP1 molecules are phosphorylated in multiple places. Some of these phosphorylation events inhibit DNA interactions, and they kick them off DNA. But they also seem to promote phase separation of the HP1 molecules by themselves. We believe there might be other phosphorylations that might do both, inhibit phase separation and kick them off DNA. So absolutely, there's a whole universe waiting to be figured out.
Can I ask something? So really great talk. I was wondering, can you do these droplets fusion experiments with nucleosome arrays rather than DNA to see how it would affect the dynamics, perhaps?
Yeah, so we've done some of those, and similar things show up. And Madeline is in the process of extending what we see here to nucleosome arrays. Absolutely. The next step is nucleosome arrays. Our goal here was to uncover what are the intrinsic properties, and then we can build in complexity. But yes, we're beginning to see something similar with nucleosome arrays. The length dependence is different because the DNA is wrapped up in histones, but it seems to be a scalable property.
Right.
Any more questions?
Gita, I have a question. This is Natalia.
Yes.
If you in vitro, if you were to reconstitute HP1 alpha with really short pieces of DNA, would you also get phase separation, or is that consistent on this trapping mechanism?
So that's a great question. Madeline has done extensive studies on that. There is a strong length dependence. So when you increase the DNA length you can get phase separation with fragments as small as 40 base pairs. But the critical concentration, the concentration at which you get phase separation with short pieces of DNA is much higher. It's only when you go to KB length pieces of DNA that the concentrations you need for HP1 to form phases start matching the biologically found concentrations of low micromolar. So definitely length matters, and for some of the reasons that you just mentioned, because the longer the DNA, the more multivalent it is, the more binding sites there are. And it's also going to be coupled. It's going to be cooperative folding enabled by HP1 molecules.
Gita, you could imagine regulation from the other side too, right? So you could have a multivalent organizer like HP1. It doesn't seem like HP1 might be that one. But you said, so if you basically string multiple HP1s together so that they can't exchange quickly between the droplets, then you could have similar condensation behavior, even with shorter pieces of DNA, perhaps.
So when you have shorter pieces of DNA, the way we conceptualize this is, if you just take a long piece of DNA with multiple HP1 molecules and you cut up the DNA, right? Now you're making something that's intramolecular into intermolecular. So when you have droplets with shorter pieces of DNA, the DNA can now readily exchange, right? But when you have long pieces, you can't. The HP1 molecules always exchange because the off rates are really literally on the order of seconds. So unless the off rates go down into the minute scale, HP1 molecules are always going to come on and off. And it's kind of the strength in numbers argument, because there are multiple HP1 molecules holding the DNA in place. That's what inhibits it from exchanging. But I'm not sure I fully got your question, Tanya. You were saying something about multiple.
Yeah, so now you have the multivalent DNA and the HP1s
Right.
that exchange quickly. You could turn it around. I don't know what that would mean biologically, but you could have a multivalent HP1 and short DNA pieces. And that should
So you could engineer it, you're saying, in principle. Yeah. If you could engineer it by covalently connecting multiple HP1 molecules, absolutely. You could do a lot of fun games by changing the off rates and adding multivalency covalently. Yeah.
Thank you, really fascinating.
Thank you.
We'll go to the next talk by Professor Tawfik from the Department of Biomolecular Sciences at the Weizmann Institute, talking about the wonders of enzyme regulation. Unmute yourself, Dan.
Okay, thanks, Noam, and thanks also primarily to Nevan, who has been really the key driving force behind this very nice initiative. And let me also first thank two terrific people who led this work, Noda Garcia, who a former postdoc, who is now a new faculty member at the Hebrew University, and Vijay Jayaraman, who is a postdoc following Lianette's footsteps. And if you have lots of emails to answer, this talk's bottom line is very simple. Keep your enzyme enemies close. So it was really Jacques Monod in the late 1930s who realized that in living cells, enzymes turn on and off in response to variations in nutrients. And also that this phenomenon is fundamental to biology, not only in metabolism, but also in development or immunity, for example. And the first outcome of his curiosity was the famous lac-operon, who became the first and leading model for regulation of gene expression. However, the work of Monod, together with François Jacob, brought in a second great discovery, and that is allostery, regulation at the protein or enzyme level, together with Dan Koshland, by additional regulatory sites that turn enzyme action on and off in response to a regulatory metabolite. But maybe nature has other additional mechanisms for regulating enzyme activity. And an opportunity to discover such a new regulatory mechanism came when Lianette began to study the catabolic glutamate dehydrogenase of Bacillus subtilis. So what GDHs or glutamate dehydrogenases do is to take glutamate and break it down to 2-oxoglutarate that then goes into the TCA, feeding in as a carbon and energy source, and ammonia, which is largely diffusing off the cells. And glutamate dehydrogenases are among the most highly regulated enzymes, if not the most regulated ones. Yet the precise mode or modes of regulation remain unclear to this very date, as hinted in the title of this review by Paul Angel, who had been studying them for 45 years. The reason for this complex regulation is that glutamate is not just one and other amino acid, but rather, in addition to being a protein building block, glutamate has many other roles as osmolyte, counterion, and various other roles. And foremost, glutamate is really a hub for carbon as well as nitrogen metabolism. Consequently, its concentration in the cell varies widely, and it can be as low as several millimolar or as high as 200 millimolar. And accordingly, glutamate is either broken, if it's in excess, via catabolic glutamate dehydrogenases, or synthesized by an enzyme that basically reverses this reaction, namely takes quetaglutarate and NADPH and generates glutamate while using glutamine as the source of ammonia. And whether glutamate is degraded or synthesized in the cell depends largely on what is the carbon and nitrogen source. So in a nutshell, if you grow cells on glucose as the carbon source, and ammonia or amino acids as nitrogen source, this would demand synthesis of glutamate. So basically, glucose would feed into the TCA through oxaloacetate, that in turn would be generating ketoglutarate that would be making glutamate. However, if you grow cells on amino acids such as adenine or histidine, or for sure glutamate or glutamine, then this demands the breaking down of glutamate into ketoglutarate and feeding into the TCA. And as it turns out, Bacillus subtilis has two catabolic GDHs. The one that caught the interest of Lianet is called GAD-B. And what's unique about it is that it is highly and constitutively expressed regardless of the carbon and nitrogen source. So it is not subject to any kind of transcriptional regulation. And in fact, Lianet initiated the interest in GAD-B and in Bacillus as she was looking for a model to study pleiotropy. This is a complex topic, and indeed she showed that 70% of the mutations in GAD-B show pleiotropic fitness effects. That is to say, they can be deleterious in one condition, but beneficial in another condition. And that makes that what we call the fitness landscape look very interesting and very different from what we are used to think. But this is a different story and now a published one, so I will not go into it. But what Lianet clearly found is that GAD-B must be regulated at the protein level. And the reason is that if you take its paralog, which is called ROCG, which is 75% identical in sequence, and you place it under GAD-B's constitutive promoter, and you grow the bacteria in glucose ammonia, condition where GAD-B should be silent because you are making glutamate, not breaking it down, then this has a very deleterious growth effect, which implies that basically GAD-B is regulated at the protein level. So our hypothesis actually was in this paper that GAD-B is regulated by association, dissociation of its hexamer. The enzyme is a hexamer, because at least in vitro, we could see that the hexameric structure depends on glutamate. However, as Vijay, who continued Lianet's project, discovered, evolution was much smarter and much more original than we were. So what Vijay did is to do cross-linking in order to monitor the oligomeric state in vivo under two growth conditions. One would be glucose ammonia, where we expect that GAD-B would be silent and would dissociate into monomers or dimers, and histidine, where I expect the enzyme to be a hexamer and be active. He did see a very distinct difference in the cross-linking pattern, as you see here, between glucose and histidine, but it was actually exactly the opposite of what we expected. The high molecular weight species were in glucose, not in histidine. Next step, he did a pull down, which revealed actually a very cool result. In glucose ammonia, and only in glucose ammonia, GAD-B is bound to an enzyme that catalyzes exactly the opposite reaction. That is glutamate synthetase, or GLT-AB as it's called in Bacillus. So what happens is, keep your enzyme friends close, but your enemies even closer. GAD-B is basically in association with the enzyme that catalyzes the opposite reaction, meaning taking alpha-ketoglutarate and turning it into glutamate. And indeed, GAD-B in complex with GLT-AB is active, yet has a 20-fold higher Km, meaning much lower affinity to its substrate. And intriguingly, in the presence of GLT substrate, specifically alpha-ketoglutarate and NADPH, GAD-B is completely silenced. The enzyme is effectively inactive until 50 millimolar glutamate, whereas the regime where GLT-AB is expressed is in the order of 10 millimolar. And further, very recent, exciting cryo-EM data of the complex that Vijay pulled down from Bacillus give us, or begin to give us, atomic details at 3 angstrom resolution of the complex, where you see the hexamer of GAD-B, the dehydrogenase, bound to 2 heterodimers of GLT-AB of the synthetase, and where the synthetase is basically bound right in the active site pocket of GAD-B. But let me go back to kinetics and to real life. When I mean real life, in the cell, we have not just GAD-B and GLT-AB, but also all the substrate and products of these enzymes. So what is happening in the cell? Let me just quickly remind that reduction of NAD would give you increase in absorbance at 340, whereas its oxidation, which happens when you have GLT-AB activity, would lead to a reduction. So when you put the 2 enzymes together with all the substrates, and which are then the products of the reciprocal enzyme, something has happened that I have never seen in 30 years of doing enzyme kinetics. Basically, you see these oscillations. GAD-B starts acting, as you see here in the increase. It releases some oxoglutarate that in turn shuts it down and allows GLT-AB, the synthetase, to kick in, as you see here. The synthetase would exhaust the oxoglutarate, which would in turn turn on GAD-B that starts to work. So you see these oscillating kinetics of suggesting or indicating that it's an OR gate, meaning either GAD-B or GLT-AB are active, but never together. So why did this complex evolve in the first instance? Well, this is what we are currently aiming to understand, because in principle, GAD-B could have been subject to transcriptional regulation in the mode of most other bacterial genes, or to ordinary allosteric regulation. Why this unique complex has evolved? And the clue seems to be in these intriguing recent observations that bacillus biofilms oscillate when growing on glutamate. And what we suspect is that the GLT GAD-B complex and these oscillating kinetics that you see here are in fact the oscillator, that they are driving these oscillations. And this unique regulation that has, by the way, very high cooperativity that cannot be achieved by allosteric regulation is the reason for, or was the driving force for the evolution of this unique regulatory mechanism. So let me thank again Leonette and Vijay, who did all the work that I presented, and a bunch of collaborators who's been very helpful, including, and I said, Jamie Fraser's group in UCSF.
Thank you, Dan, for amazing talk. Really fascinating. Are there any questions? So I need a clarification. The GAD-AB, when it's in complex, is it active?
It's active unless you put the substrates of the other enzyme, or the synthase. Okay, if you put the moment you add the substrates of the other enzyme, it shuts down. It's in effect, I mean, it has a high Km, but it is more active than when it's on its own.
Any other questions? Okay, so if there are no more questions by the time I finish talking, so we'll go for a 10-minute break, which means we'll gather again at 7:20 p.m. Israel time, which is what, 9:20 a.m. Am I correct? I think so. Okay, so yes. See you in 10 minutes. Dan, if you're still here, there is a question. don't know if you're here.
I'm here.
Have you tried native mass spectrometry for the mixer?
Sorry, have we done native?
Native mass spectrometry for the mixer.
Yeah, we have. Actually, I didn't have time to show it with Michal Sharon. So there, yes, we see actually the complex seems to be heterogeneous. The GADB is always
9:20 a.m. here, and some other time, also 20 minutes past in Israel. So our next speaker is James Fraser from QBI UCSF, and he's going to be talking about a question that many people have thought about quite deeply. So I'm curious what this is going to be. What happens when a protein binds a ligand?
Thank you. It's a real pleasure to be speaking to this audience. I'm going to talk today about work that we're doing as part of the QCRG Structural Biology Consortium, and then also a brief story that a graduate student, Stephanie Vankovic, has produced recently that I think will provide insight into how we're going to further that work. Before I get going, I just want to acknowledge that the Mechon Weizmann is like a very special place to me. I visited for a sabbatical in my graduate work with Danny in 2010, and then here we are in almost 2020. Unfortunately, these days, it's only in this form. We don't get to go hiking anywhere nice. We just go on Zoom. But it's wonderful to see this connection between, I think, two very special scientific communities, UCSF and the Weizmann. So I'm going to start with just the problem that we currently have no effective small molecules therapies to stop the spread of SARS-CoV-2. We have some treatments that may help the late stage symptoms, but we really lack the therapies that we can deploy on a grand scale. And this is a problem that many have realized. And even though a lot of efforts may not impact SARS or COVID-19, a lot of us are interested in preventing the spread of the next outbreak, which will inevitably happen at some point in the future. And so here we've gained a lot of inspiration from our friends at Oxford and as well near London at the Weizmann, who've been attacking the main protease with fragments through crystallographic fragment screening and electrophiles, and also really galvanizing the international computational chemistry and drug discovery community in this spirit of openness, which I find really amazing. And so at UCSF, we got interested in a less well-studied SARS target, the macro domain of NSP3. And we got interested in it because it has this antagonistic relationship with the sort of PARP family of enzymes that Alan Ashworth studies here that post-translationally modify proteins by tagging ADP ribose onto them. And as well, Brian Schoiket was very interested in it as a docking target. So we've recently established here at UCSF a really nice fragment soaking pipeline for crystallography. We have a couple libraries, and we have a really nice sort of pretty high throughput system for crystallizing, soaking ligands in using acoustic liquid handling, vitrifying them in a sort of higher throughput way than normal, collecting data and analyzing the data. And as part of this coronavirus consortium that many of us are a part of, we put a big effort into optimizing this macro domain enzyme for identifying new ligands via fragment screening. It's a real problem, especially for macro domain, where we have no synthetic ligands for it. So we really have no starting point for drug discovery. And very quickly, we got beautiful diffraction data to about 0.8 angstrom resolution. We can really see the individual atoms and hydrogens. And when we screened our fragment library, we also got a really nice data, although a bit of a problem in that we had multiple crystal forms. And this was really a huge team lift from the whole QCRG community, spearheaded by a lot of these folks here that I'm calling out specifically, especially Gail and Corey. And as we were wrapping it up, I got in touch with our friends at Oxford and the facility there, because I know Frank very well, and they had been doing something very similar. And so we decided to combine forces and release the data together. And what you see on the left of the slide are the fragments for the macro domain arrayed across the structure from their initial soak. They had a lot in the active site, which hopefully you can see here, and then a much smaller contribution from our initial effort here, where we had about 13 fragments bound. But after sort of mining them for information, we found that they had actually a slightly different crystal form, and we were able to get then a much higher fragment hit rate with our next iteration. These results just came in over Slack as I was preparing this talk, so I'll just zoom in here on the time. So Galen is literally working around the clock. This came in at 4 o'clock in the morning, California time, where we have these many new hits. And as well, very excitingly, a whole bunch of hits that emerged that confirm docking predictions that Brian Scheuket and a postdoc in his lab, Stefan Gabauer, had made. And so these are giving us really high resolution insights into how to bind ligands to this relatively understudied SARS target, which we think is a really compelling target. And so now I think really the hard work begins. You know, how are we going to improve these hits? And this is where I want to transition the talk to something a little bit more theoretical and maybe esoteric. I really want to reframe this question to like think about what is missing from current approaches that meld medicinal chemistry and structural biology. And so when we think about the goal in these types of structure-based drug design or ligand design projects, we're really trying to optimize the free energy of binding. But most of our analysis when we open up Pymol or Chimera is really focused more on our intuitive understanding of the enthalpic parts of this equation. Really, we're thinking a lot about, you know, hydrogen bonding and optimizing van der Waals surfaces and things like that, which we could categorize more as the enthalpic part. If we start to think about the other big contributor, the entropic part of this, you know, we can break it down into the protein component, the ligand component, the solvent component. The solvent component we understand pretty well. The ligand component, you know, mostly thinking about rotational and translational degrees of freedom here. But the protein component is actually really fascinating to me, where we could start to think about, does the protein flexibility change? And in particular, does the side chain flexibility of proteins change upon ligand binding? And there is, you know, one paper here that's probably one of the more cited, but actually never read papers. A lot of people cite this paper without reading it. I'll point people here that has this idea of allostery without conformational change. And the idea is sort of illustrated here where, you know, we're looking at changes where a side chain, you know, might move from not flexible in one state to very flexible in another. And indeed, Josh Wand has pushed on this concept to draw a general trend between side chain order parameters, an NMR measure of flexibility, and entropy measured through calorimetry in several systems. And Babis Kalodimos has also pushed on this. And what we're starting to see is these really interesting patterns where protein flexibility changes across the protein. So what Stephanie wanted to ask is, can we extract some general principles of how protein dynamics change upon ligand binding? And, you know, we could think of several models where this might happen, you know, where the protein might rigidify or loosen in certain areas. What she did was went to the PDB and assembled a really nice data set of as close to perfectly possible match pairs as possible. And we're crystallographers by training, and so we're really paying close attention here to try to control all the external differences. And I'd be happy to talk about that more in the questions. What she found when we started to just count the number of alternative conformations, the number of times a residue was modeled as being mobile, we saw something that was totally bizarre, which is that proteins appeared to become more flexible, less rigid near the binding site after binding a ligand. So rather than sort of this model of enthalpically driven binding where things would tighten down, we got the complete opposite. So, you know, this led us to, you know, sort of favor a model that proteins become less rigid upon ligand binding, which was the exact opposite of what I expected going into this. But there's another model, which is that crystallographers only pay attention to modeling stuff near ligands. And to test that model, we used some software that we've developed over Henry van den Bedum, a very close collaborator, that allows us to take electron density and automatically fit alternative conformations into it. And this has less bias, I would argue, than what crystallographers are currently doing. And at the very least, I could say it has a consistent bias that will allow us to ask these types of questions. And so when we ran through these very tightly matched pairs, we saw an interesting change, which is that there was no change in the number of alternative conformations when you compare the apo to the bound structure, but that every other measure, every other proxy for entropy that we could come up with changed in a very characteristic way, where near the ligand, it tightened up. The B factors decreased, the flexibility between alternative conformations increased, measures that we have to summarize those things decreased and indicated more flexibility. And far from the ligand, we saw the opposite, things loosened up. And so this result was sort of, we're hitting on it in many different ways. It's very highly significant, and we're trying to falsify it, but we're starting to come up with a general pattern where proteins become more rigid around the ligand binding site. But there's this decrease in rigidity, this increase in flexibility elsewhere. So what I told you about today is how we think the macro domain is a really compelling target, and we're identifying the first synthetic ligands. And when we go forward, I think the challenge is really to integrate our ideas from sort of this basic science study, where we see this tightening of local conformational heterogeneity that may be entropically compensated in distal regions. And so with that, I'll thank the folks who put in the hard work here, a lot of great collaborators that I acknowledged along the way, at least textually, especially the folks in my group who did the work. And I'll highlight especially Galen, who just worked so hard on the macro domain stuff, and Stephanie, who's had these really cool bioinformatic results. And with that, I'll take any questions.
Thank you, Jamie. Super neat. Are there any questions? Maybe I can get us started here while people are typing, hopefully. So this finding that other regions become more flexible upon ligand binding, can you in any way sort of stratify this based on, is there a conformational change in the binding site upon ligand binding?
Yeah, so that's one of the sub-analyses that we have planned to sort of look at the idea of conformational change. This data set is dominated by what I would call pretty highly pre-organized binding sites. And some of that's probably a bias of it being a crystallographic data set. I would guess that our data set will be much larger for the things without conformational change. But it will be super interesting to sort of break it out in that way.
Yeah, and it's sort of similar models again, right? Is LSTOR mediated by actual visible conformational changes, or is it just all in fluctuations?
And the few examples we've really been looking at in detail, it is pretty subtle the way the tightening up in one area can lead to this dance in the distal region.
So this,
right. Here's a question from Zaki. Sorry. Can you show that loosening distally contributes to delta G?
Yeah, so that's a hard thing to show. I have some ideas about how we can do it, things like cross-linking or getting together with Tanja and trying to get her to sort of tighten the packing in distal regions so they can no longer respond, and then showing a change by, in binding affinity and things like that. We haven't done those types of experiments yet. Obviously, that's the thing that we want to push on in sort of isolated examples. And I think the macro domain is a beautiful system for trying these things out.
Can I follow up?
Yeah.
So I appreciate that it will be hard to cross-link and see that it affects binding, but at least computationally, can you estimate what's the extent of free energy that is being gained by this loosening distally, and to see whether it's appreciable compared to the delta G of the interaction?
Yeah, we could do that. And my guess is it will be on the order of, KT, a few KT. It's not going to be the difference between, picomolar and micromolar, but it is going to be an important aspect, I think, especially for driving specificity or for just getting over the hump, of a certain cliff in free energy of binding optimization.
Thanks.
Yeah, so
another
I also asked comment that it could go both ways, right? It could increase affinity,
Yes,
or decrease it.
I should say modulate. Yes, my wonderful host Danny, points out you can go in either directions. I'd say that the overall trend is what I indicated: this tightening and then loosening. But I'm sure we'll find examples that go the opposite.
Yeah, you could think that it would break down some interactions, so the overall energetics of the system would be lower, or yeah.
And you can get trade-off between enthalpic interactions and entropic components that are going to be hard to sort out, but really important. Okay, so I think we need to move on, but Jamie, there's a couple more questions in the Q&A, so maybe you can
I will do my best to answer them.
All right, so our next speaker is, hopefully I pronounce your name correctly, probably not, Efrat Shima.
Very good.
From the Weizmann Institute Department of Biological Regulation, and she's going to tell us about single molecule epigenetics. For today's session, and that's my UCSF QBI colleague, Lani Wu, and she is going to tell us about single cell cancer fate decisions.
Thank you very much for inviting me here. I want to say everything I'm going to talk about is going to be joint work with Stephen Altshuler. So he's on my side to answer any questions too. His hands is there. And our lab have been focusing on understanding cell decisions. We study the cell decision in tissue and normal tissue and also disease tissues. And the model systems we have right now in the normal disease tissue as the gut epithelium, neuronal circuit, as well as recently we just started a new project on studying the responses of tissue in hypoxic stress. In disease, we study cases in gut toxicity, neurodegeneration, and cancer. And today I'm going to focus on our study in cancer. In the past, we have studied cell decisions on how cancer cells can make whether they're going to die or become tolerant in high doses of drug. Today, I'm going to take you to a story in non-lethal doses of drug where cancer cell can make the decision to stay proliferative or become senescent, as well as a new study by a graduate student, Leanna, and she studied how cancer can make the decision to become more stem-like cells or differentiated. And to start with the first study in terms of how cell can make decisions in non-lethal doses of drug, I want to get you familiar with the concept of senescence. So for example, in this picture here, you see A549 lung cancer cell line. In DMSO, they look like this, but when you treat them with drug, in non-lethal doses of drug, they become senescent after maybe 5 or more days. And this phenotype can only be, currently, the best marker to look at it is beta-galactosidase. And the cells become still alive, but they have kind of enlarged morphology and they kind of have very different state. And in principle, this should be a permanent cell cycle arrest. And the question that the graduate student, Charles, trying to study is that if you treat the cells with a pulse of drug treatment for 1 day, and then remove the drug, and after 5 or 7 days, they can still become senescent or remain proliferative. And the question here is that with the current study that you have to wait until 4 or 5 days after the drug treatment, that it's kind of late to really detect the decision. So the question he has is that, can we predict the cell fate decision earlier And if you think about it, with drug-induced senescence, what we know about today is really a DNA damage response. And one of the key readout is P21. And under the DNA damage response, P21 is going to be overexpressed. And it could stop the cell cycle and cause the cell to become senescent. And so in our study, when we do the 1-day pause of drug treatment, if we look at them after 5 days treatment, 5 days after treatment, we see that the cells become senescent, and that is being read out by the P21 expression. So we see two subpopulations of cells, one with high P21 and one with low P21. And the cells with low 21, they look like they have been proliferated. And the cells with high P21, they really exhibit the senescence phenotype. And if we also look at early during the drug treatment, we also see two subpopulations of cells, one with high P21 and low P21. So a naive hypothesis would be maybe the cells with low P21 early are the cells that would become proliferative. And the cells we have high P21 early should be the cell that become senescent. So, however, this seems to be a simple hypothesis, but no one in the field has really tested that. So to really do this, the only way we can think about is by lysol tracking. So Charles tagged the endogenous P21 with green color, and then also labeled the cytosolic with a red color. And with this, he was able to get time-lapse microscopy and started to track cells, And so this is essentially the main tools of this study. So you can see that we can track cells in time during the drug treatment and after the drug treatment. And you see some cells with high P21 pulse. And interestingly, after the drug treatment, the P21 expression goes down. And because we can, with the lysol imaging, we can track them through division. And for this particular cherry pick cells, in fact, that they go around, well, after the P21 drops, the cells started to go through cell cycle division. And we also see cells with really low P21 early on, but they increase the P21 high, but they don't really divide. So with this, we decided to really gather some statistics. So we created a heat map of P21 intensity across time. And we also make a very practical decision here. We're going to cause cells to be senescent if they have never divided once during our observation time. And then if they have divided more than once, then we say that they are proliferative. And with this, we were able to label the cell, assign cells with senescent cell fate or proliferative cell fate. And with this, we can kind of gather the average response dynamics. So we see that for cells that with proliferative fate has an early pulse during the drug treatment, but with lower P21 expression. And interesting, the cells with low P21 expression early on, they never divide and they go have a P21 high expression towards the end, and suggests that our simple hypothesis was completely wrong. So to really make sense out of this data, we decided to also build the associated cell cycle dynamics for all these cells that we tracked. With that, we are able to reanalyze the data based on the longest cell cycle phase in drug. And what we have found is we found 3 different patterns. It turns out that the cells that have been stayed the longest time in S and G2 cell cycle phase, they have a very canonical response that they have low P21 during the drug treatment, but the P21 would increase after the drug removal. And then the other one is that for the majority of G1 cells, they have an early P21 pulse, but the P21 expression would drop and then it stay low during the proliferation time. And interestingly, we also saw a subpopulation outside with G1 phase, but they have prolonged much higher P21 expression for the whole time. With this, we decided to evaluate, so we find that there are two paths to senescence and one path to proliferation. With this, we decided to reevaluate one of the hypotheses in the field that we can increase P21 expression and to cause the cell to push the cell into the senescence state. But our data suggests that if we push the P21 high, it will somehow maybe even bring the cell into the proliferative subpopulation. And so that may not be the right thing to do. So we decided to treat the cells with Natron 3A to cause the cell to have higher P21 in different degree. And we see that in terms of proliferation, we see a Goldilocks behavior and suggest that the drug treatment by increasing the P21 may not be a good idea. And this kind of mis-sulfate decision have been seen across cell lines, across different drug treatment, and the Goldilocks zone is also have been observed. And this suggests that to really study the sulfate decision, we really need to take into account the dynamics and the heterogeneity. And then the last story I'm going to quickly take you through is mutation. In cancer, we observe many different mutations. And some of the mutations, we know how they affect the proliferation directly. And some of them, we can even understand how they affect the drug treatment. But many of them, we still don't know anything about it. Especially in AML, we have a very clear hierarchy of sulfate decision. And in this case, we also see a relatively few genetic mutation. But even in this case, we don't really know what every mutation does, especially in the TET2 case. TET2 is a DNA demethylase, and there's no obvious genomic target. However, we see them often before drug treatment, during drug treatment, and also during relapse. And the question is, how does TET2 confirm fitness? And so we developed an isogenic cell line. And we studied it, and we did not really find anything concrete until we find that these cells have some different proportion of stem cell and differentiated cell. And for example, if we sort the cells into more pure population of stem cell and differentiated cells, and we observe it across time, and we see that the TET2 mutation, that when the stem cells really have much less differentiated cell compared to the wild type, and if we look at the differentiated cells population, the TET2 mutant has much higher stem cell level and suggests that they have a different type of switching behavior. The TET2 mutation cells have higher chance to switch to the stem cell-like state, and which is a more proliferative cell. And that's how TET2 mutation confirm fitness. With all this, I would like to thank my lab and the two grad students that created these two projects. Thank you.
Thank you, Lani, and by extension, Steve. There's one question here already. Do P53 oscillations have anything to do with the different classes of P21 expression?
We're not sure. The only that was a hard question because we don't monitor P21. P53 in live cells, so we don't have those resolution.
I think one thing that I was wondering, you chose a specific duration of the doc's pulse, but I'm wondering whether you tried perhaps other durations of the pulse and whether that has any effect on the different outcomes that you see.
So we did try different ways. So for example, when we treat them with a whole 5 days, and essentially every cell becomes senescent. And so we just kind of pick a time so that we have a reasonable percentage of cells after all the division that we can still track them.
Okay, here's another question. Thanks for your talk. Have you checked what happened if increasing P21 expression in specific times, such as in later stages?
Yes, so the fact that we were doing, sorry, one more time.
I guess that, yeah, so the question is about, so you had that experiment where you increase P21 expression, but you could also do that at different times during the experiment, in particular at later stages during your experiment. Have you looked at that?
As we did not do all the lifestyle tracking for that particular case, but we did a little bit of test in the beginning trying to figure out the condition. And with fixed out assays, we did not really see much difference.
Okay, thank you, Lani, and thanks to all the speakers today. I, again, had an absolutely fabulous time learning about exciting and really creative and innovative and new science. And I think Nevan is going to finish this session with a few words.
Yeah, just to reiterate what Tanja was saying, just fantastic science these last 2 weeks. These have been my favorite Zoom meetings over the last couple of weeks listening to this. And it's so synergistic, right? The science in between the two institutions, and so relaxed. I love the discussion with Denny at the break. I'd like all my Zoom symposium to have a discussion with Danny at the break. We could implement that in the future, I think. So we're going to try to continue this momentum next week. We're going to have a meeting with the PIs at 8 a.m. here. I don't know what time it is in Israel. Same time, same starting time. We'll discuss how best to go forward. We had Andy Weizmann on last week, who is based in the Bay Area, but works for the Weizmann Institute. And he works on securing funding for collaborations with the Weizmann. So I'm not sure if he's going to be on the call next week, probably not. But the next step is to say, okay, what can we do? How can we frame this? And then we'll probably go back to Andy Weizmann and ask him to help us raise some funding for whatever we come up with. And who's invited? I mean, it's the PIs that spoke, but if any other PIs want to get involved, just contact us. Obviously, the more the merrier, and then we'll discuss a little bit about the itinerary and let the people know beforehand what we're thinking about next week. So that Saki, Danny, Noam, Tanya, and I will discuss how we think we should frame it, and we'll let everybody know. So, Saki, did you have something?
Yeah, just to say on behalf of the Weizmann people, thank you, Nevan, and all the administrative team at QBI. Thank you, all the speakers. It's been really exciting. Thanks for all the participants. I hope you enjoyed us. You enjoyed it. It was really stimulating, and we should do much more together. That's obvious.
All right, great. Thanks, Saki. Danny, did you have something?
No, just thanks, everyone.
Okay, Tanya and Noam and Saki and Danny, thank you very much. And we'll see hopefully many of you next week at the same time.